Welcome to STEMinism Club.
Hello everyone and welcome! Today we are going to be trying an experiment that extracts the DNA from strawberries. So first we're going to give you a little bit of background. So your body is made up of little spheres called cells. These spheres are very small, less than the width of a strand of hair. Each part of your body is made up of different types of cells. Your skin is made up of skin cells, your brain is made up of brain cells, and so on. The question is, what tells your skin cells to act like skin cells or your brain cells to act like brain cells? This is where DNA comes in. Every cell in your body has 2 copies of your DNA. Every person, except for identical twins, has unique DNA, which is why we don't all look the same. The DNA tells your cells what to do. For example, in a skin cell, your DNA tells the cell to act like a skin cell. You might be wondering why we're using strawberries for this experiment. Well, there are a couple reasons why they're the best choice. Firstly, strawberries contain more DNA than most fruits. They are octoploid, which means they have 8 sets of chromosomes. Human cells are mostly diploid, meaning they have 2 sets of chromosomes, so strawberries just have more DNA overall, which makes it much easier when you are trying to extract it. The second reason is that ripe strawberries contain 2 enzymes, a type of protein, to help break down the cell walls, making it easier to extract the DNA. Each ingredient in this experiment has a purpose. The dish soap dissolves the cell walls of the strawberries so the DNA isn't trapped inside. Salt is added to release the DNA strands by unclumping them. Finally, similar to oil and water, DNA and rubbing alcohol can't mix, so when DNA and rubbing alcohol are added together, the DNA floats to the top. Okay, let's begin. Before we start, make sure that you have all of these materials so that you don't have to scramble for them later. So the first thing that we're going to do is we're going to take our 2 strawberries and remove all of the green leaves from them. And after that, what we're going to do is we're going to take the strawberries and put them into a Ziploc bag. We're going to try and make sure that we have as little air inside as possible so it's easier to smash the strawberries later. And we're going to seal the bag completely to make sure that things don't go everywhere. We're going to smash the strawberries for 2 minutes really thoroughly to make sure that we're releasing all of that DNA. So this is me smashing the strawberry for 2 minutes. Make sure it's 2 minutes. So after that, we're going to take a glass and a teaspoon, and we are going to fill the cup with 1 teaspoon of salt, and then we're going to take our dish soap, and we are going to put 2 teaspoons of that inside with the salt. After that, we are going to take 1/2 cup of water, so make sure that you have something that's made for liquids to fill up with water. We're going to pour that into the dish soap and salt mixture. After that, what we're going to do is we're going to just take a spoon and mix it around for a little bit. Make sure that it's thoroughly mixed. So then we can set that aside. And we're going to take our strawberry mixture that we just smashed, and we're going to take those teaspoons. We're going to take 2 teaspoons of the mixture that we just made. This is the extraction liquid. And we're going to put it in with the strawberries. So again, we can set the mixture aside now. We're done with that. We're going to take the strawberry Ziploc bag and close it with as little air as possible. So make sure you're squeezing out all of those things. And we are going to basically mix that in. So we're going to smash it back into the strawberries for 1 minute, trying to keep as many bubbles out as possible. Next, we're going to take another glass, a separate glass, and take a coffee filter. Your parents should probably have one of these. And we're going to put the coffee filter inside of the glass. And this is basically to separate the solid parts of the strawberry away from all of the other parts. So you're going to pour in that strawberry mixture, and you're going to try and squeeze out all of the liquids. Make sure that it doesn't break. We want to keep those solid bits separated, so we only want really the pure liquid to come out. It might be a little bit difficult, but you should get it eventually. So now that we have this strawberry liquid, we're going to take our rubbing alcohol and pour about exactly as much alcohol in there as there is strawberry mixture. So it should be 2 times the volume as it was before. So then after that, that should actually do the extraction of the DNA itself. So you don't want to mix this the strawberry mixture at all, because now you can see that you can take like a chopstick or a fork or something and pull out this DNA. So basically it collects at the top once you add that rubbing alcohol, and so basically you can pull it out. And Look at that. It's the little stringy bits, and that's what it looks like. In case you're wondering what it looks like on a microscope, this is what it looks like. So if you just put it on a slide and sort of move it around, I'm not totally sure what this is, and I think that could be a fun little exploration just to see what you notice and wonder. But yeah, that's a really cool way to look at what's happening. We obviously can't see the DNA itself because it's too small, but you can see definitely the bubbles, and so this is what the final apparatus looks like. Good luck, everyone!